First speaker is Professor Ranbir Das, who is fondly addressed as Rana. And Dr. Ranbir Das is an expert in structural biology, biophysics, biochemistry, and host pathogen interactions. His group studies interactions between viral proteins and host proteins by combining multiple techniques involving NMRS structural biology, cellular biochemistry, and virology. Dr. Das graduated from the Indian Institute of Science under the mentorship of Professor Anil Kumar. Dr. Das has worked as a postdoctoral fellow and a staff scientist at the National Cancer Institute in the laboratory of Professor Andy Bird. In 2014, Dr. Das established his research program at NCBS TIFR, Bangalore. Several pieces of original work by Dr. Das has been published in Molecular Cell, Structure, eLife, EMBO Journal, Journal of Molecular Biology, etc. Now I request Rana to take the forum and start this talk. But before that, I'll appeal to everybody. If you have any questions, please raise your questions in question and answers. And in the chat box, we will revert back to you later. So Rana, over to you. Next 30 minutes is yours.
Thank you, Professor Surya Prakash. I thank you all for attending this webinar. I hope all of you are staying safe and healthy. I congratulate NMRS for organizing this fantastic webinar series. It uplifts our spirits by exchanging data, which is currently very hard to travel and attend conferences. So this is the perfect way currently to get some inputs from our recent data and think the way ahead. So today I'm going to speak about a couple of techniques that we recently developed to monitor protein ubiquitination in real time by NMR. So as Professor Surya Prakash said, the interest of the lab is to study how viruses manipulate the host cellular pathways. You see, the virus, when it enters the cell, has to undergo a variety of steps, starting from viral entry, uncoating, replication, making viral proteins, and then assemble more virus particles and egress out of the host cell. Now, to do this, the virus manipulates a variety of cellular pathways and cellular proteins, host proteins, to do its function. The current pandemic has really underlined the broad gap that we have in understanding how viruses modulate the cellular pathways, and this is something that we are trying to understand. So as you see, the virus comes with a very minimal package with a minimal DNA or an RNA, viral RNA. And it also has to utilize a variety of cellular mechanisms to modulate the cellular pathways. So it utilizes transcription, translation, which is the making of the protein from the RNA. And at the end of the translation, there's another interesting step that happens is called post-translational modifications, where moieties are attached to the protein, which changes their function, localization, or state. And as the viral proteins or the viral viruses exploit the transcriptional and the translation machinery, they also exploit the post-translational machinery for its advantage. And this is because if you look at the human proteome, we only have 20,000 genes, but due to alternative splicing or alternative promoters, the resultant transcription is around 5-fold higher than that. Added on top of that, there are post-translational modifications which happen on the protein, and now the proteome is diversified by almost 10-fold. So post-translation modifications is a very effective way to increase the function or the state of the protein. And this is something that the viruses also exploit for effective viral replication. Post-translational modifications, or the modifications of proteins, can be a variety of ways. They could be small molecules like phosphate groups in phosphorylation, which are attached to the proteins, or they could be slightly bigger groups like entire protein molecules, which happens in ubiquitination and sumoylation, which is attached to the proteins. Now, by a series of reports in the last couple of years, we have shown that viruses not only exploit the post-translation modifications, but there are crosstalks between these post-translation modifications, which are also exploited by the viruses. So there could be crosstalks between sumoylation and ubiquitination, phosphorylation, sumoylation, and all of these crosstalks are actually exploited by the viruses. Now, as we expand to study this further and understand more about the mechanisms of how these post-translational modifications are exploited, the need of the hour is to develop better tools to study PTMs. And this is something that I'm going to talk about today. And the modifications that I'm going to talk about today are the two modifications, ubiquitination and sumoylation, where small proteins, ubiquitin or sumo, are attached to the substrate protein, and that changes the function of the substrate. If we look at the enzymatic reaction of ubiquitination, you would see that it is categorized by different classes of enzymes. There are three classes of enzymes, E1, E2, and E3. And they, in a coordinated fashion, assembles these ubiquitin chains on top of the substrates, which I am showing here. Post the conjugation of these ubiquitin chains onto the substrates, they they undergo a variety of functional changes or changes in localization. But one of the most studied effect of the ubiquitination is the degradation of these protein substrates by proteasomes. I'm going to talk about the other functions of ubiquitination that also changes the function of the protein substrates. But briefly, I would also like to mention an added layer of complexity present in the human cells is that there are de-ubiquitinating enzymes, or enzymes that can also cleave and relieve the protein substrate from these ubiquitin chains. And we'll talk about them a little bit later. So now let's talk about the ubiquitin chains on the substrate. Ubiquitin has lysines. There are several lysines present in ubiquitin, and there is a N-terminal methionine, which is of course present in all proteins. The chains that are formed are actually formed between the side chain of the lysine ubiquitin and another ubiquitin C-terminal tail. And I'm showing the ubiquitin isopeptide bond that is formed between the lysine side chain and the C-terminal of the next ubiquitin. This is how these chains are formed, and these chains can be extended. Depending on which one of these lysines are used to make these chains can have a different varieties or specificities. They could be, as I mentioned, there are 7 lysines and 1 N-terminal methionine amine residue, which is free, and all of these chains can be formed. Now, these are what is known as homotypic chains that have a single flavor, that is only one lysine is being used to extend the chain in these cases. And what is interesting is that even branch chains or heterotypic chains where there are mixed linkages are also possible. Now, the other interesting aspect of this kind of ubiquitin chains is, as I mentioned, each chain has a different specificity or the linkage. And depending on the linkage, the function of this substrate can vary. For example, the K48 type of chain can, if conjugated to a substrate, undergoes protein degradation. The K63 type chains, when conjugated to a substrate, undergoes participates in DNA damage response and also the innate immunity of the host cell. So it is very interesting in the ubiquitin field to try and understand what defines the specificity of the ubiquitin chains. How are these chains formed specifically using a single lysine per chain in the protein substrate, on the protein substrate. To understand this mechanism of how could these chains be specific, we have to look at the ubiquitination reaction and the particular step where the ubiquitin is transferred from the E2 enzyme onto the protein substrate. And if we look at this step very closely, this is where the ubiquitin from the E2 enzyme is transferred to the ubiquitin protein substrate. And this whole thing is catalyzed by a third class of enzymes called E3, which catalyze the transfer of the ubiquitin onto the substrate and also binds to the protein substrate. So if you like try and think about, let's try and think about how these linkages could be specific. How could specific lysine type linkages be formed? So let's assume that there is a substrate which has, using one of its lysine, is conjugated to a one ubiquitin molecule. And now this ubiquitin molecule is going to start forming specific linkage types using specific only one lysine among the 7 lysines present in this molecule. I'm going to introduce a nomenclature here that we commonly use in the field of ubiquitination, which is known as acceptor ubiquitin and donor ubiquitin. Acceptor ubiquitin is the ubiquitin that accepts the ubiquitin, and that's how the chain is extended. And the donor ubiquitin is the one that is donated on the acceptor ubiquitin. So that's the nomenclature that we use. I wanted to use it repeatedly throughout the talk, so I wanted to introduce it here. So this is how the lysine from the acceptor ubiquitin then attacks the E2-UB conjugate at the active site. And then as the reaction proceeds, the diubiquitin is formed onto the substrate. So this is how the chain elongates. Now, if you think, why would this chain have any specificity and why would only a single lysine be used from the acceptor ubiquitin? You would assume that there must be specific contacts being formed between the active site and the lysine or the region around the lysine of the acceptor ubiquitin that allows this lysine to attack the acceptor site and not the other lysines that are present throughout this ubiquitin molecule. So there must be specific contacts between E2 and the substrate that would determine the lysine specificity. And so how could we study that? How could we confirm that and understand what are the specific contacts? There are two possible or two favorite ways that people do this in the field. Is one of them is to use some sort of a chemical linker that allows you to stabilize this intermediate. Because this is an intermediate, it is very transient and it's very hard to trap this thing and solve its structure. So one of the techniques that can be used is to use some sort of a chemical linker that will form this, stabilize this, which is not going to be the natural linker, but something close by. And that would help you to solve the structures of these and then determine what are the contacts between this lysine and the active site. The other approach could be to model these structures in silico and go for these structures which are the lowest energy structures to understand what could be the contacts between E2 and the ubiquitin. However, in both these cases, you have to validate them by some sort of ubiquitination assays, which will help you to validate whether these contacts that you observed are in fact what is happening in nature. So that's common. We can do these assays. We have done this before and several labs have done. For example, we can have E2 ubiquitin with one flavor and add another ubiquitin with another flavor, and then see this diubiquitin formation, which is a typical enzymatic reaction where the product is diubiquitin, and you could see that we are monitoring diubiquitin here in gel. However, these methods have some drawbacks that we have to take multiple allocates at multiple time points, and this could be error prone. So a real-time method would be much more elegant and less error prone. And we were thinking that NMR has been extensively used to monitor reactions in real time. And one of the major advantages of NMR is that it is tag free. You don't have to put tags onto any one of the molecules, which we need for other techniques like fluorescence spectroscopy, etc. So I just wanted to mention that there has been a lot of work of monitoring chemical reactions, biochemical reactions in real time by NMR. There's been a lot of work also done to monitor post-translation modifications in real time by NMR. Most seminal work's been done by Phil Salenko's group in Israel. However, there are very few methods to look at ubiquitination in real time by NMR. So our question was, can NMR be used to measure the ubiquitination rates in real time? So the phenomenon that we utilized here is very simple. We look at the lysine side chains and its characteristics, how it changes upon ubiquitination when an ubiquitin is attached to it. So here I'm showing the lysine side chain of a ubiquitin molecule, which also could be the substrate. And these lysines, they are exposed to the solvent and exchange rapidly with solvent, making them almost invisible in physiological conditions. And Junji and Marius had shown before that if you go to lower pH and at very low temperatures like 2 degrees centigrade, it's possible to observe these side chains, reduce their dynamics and the solvent exchange and observe them. However, these conditions are not conditions where ubiquitination reactions can be carried out. So we cannot look at them at these conditions. However, when an ubiquitin conjugation happens and a diubiquitin, for example, is formed, then these amine groups become an amide group, as you see in here, because the C-terminal end of the next ubiquitin is attached to it. Consequently, the spectra of this amide group changes. It comes to the normal amide region, which is around 188, 128 ppm in the N15 dimension. Earlier, it was 33 ppm. And this lysine amide group exchanges much more slowly with the solvent. And so these amide groups can be detected and monitored. So our techniques are very simple, is to monitor this new emerging amide resonance coming from this lysine side chain because its intensity is going to be proportional to the product, which is the diubiquitin in this case, or ubiquitinated substrate. So we have suggested 3 simple methods. One of the methods is, the first method is to use N15 acceptor ubiquitin as a substrate. In this case, acceptor ubiquitin, but one can also use it as a substrate and just repeatedly collect N15 HSQCs. So when you do that, when you carry out this reaction and collect repeated HSQCs, you see at 0 minutes, there is no peak in here. And at the end of the reaction, we see a new emerging peak, which is coming from this amide, lysine amide side chain. If I just keep taking cross sections of this over time and measure the intensities, I do have the an emerging peak coming up with time, and the intensities of this can be monitored over time to get the reaction rates for the formation of diubiquitin. This, as I mentioned, E3 is a catalyst. So we have done this in the absence of E3 and the presence of E3, and we see a rapid jump in the ubiquitination rates, which has been also reported by others, but this has been done in real time using NMR. The time resolution of this experiment is 1 hour on the conditions that we used. And this is what we are wondering. So this method is very simple because it uses a standard isotope labeling and conventional 2D HSQCs. However, the drawback is that the time resolution is low. It's a 2D experiment. And the second drawback is that this is now coming into the amide resonances area. And if the substrates, one of the amide resonances overlaps with this new emerging amide resonance, then it is going to be difficult to measure this amide resonance and get the rates of ubiquitination. So the alternate method is something where we used a other labeling, which is the C13 labeling on the donor ubiquitin. So now if you label the donor ubiquitin with C13 label and the acceptor ubiquitin with N15 label, then upon the ubiquitination and the product formation of diubiquitin, you have this HNC13 moiety formed, which is not present in either one of them. And so you have an amide bond that is bonded to a C13 atom. So now if we carry out a simple C13 and 15 edited proton detected 1D, which is nothing but a simplified version of HPO without any of the evolutions in N15 or C13, then you would see it, and this is the flow of the magnetization throughout the spectra. And if you carry out this reaction and keep monitoring, taking 1Ds of this C13 and N15 edited 1Ds, you would see a new peak coming out that is coming out from this moiety, which is the amide bonded to the C13 carbonyl atom. The third method uses another specific labeling where only the N-eta lysines are labeled, and there are isotopes that can be used where N-lysine is labeled only at the N15 eta, and if you grow the protein using that, only the N15 etas are going to be labeled. And one could also use this as the third method, where using the N15 eta lysine as the acceptor substrate ubiquitin and carrying out the reaction, you form diubiquination. And this is the only amide now which is N15 labeled. So if you carry out N15 edited proton detected 1D, which is again a simplified version of HSQC, you would see a single resonance coming from this amide bond. Remember that the amines are coming at 33 ppm and they are not visible in these physiological conditions of pH 8, 7 or 8. However, this amide is formed when the diubiquination is formed, and this amide is the only resonance that is present in the spectra, which will allow you to understand and monitor these diubiquination rates. So again, we see a drastic effect of the absence and presence of E3 on the rates of the ubiquitin formation, and the time resolution of this experiment is around 6 minutes. So this is a much better time resolution compared to the 2D experiments. So these are the three methods that we suggest. And now I would like to go ahead and see how using these methods we could understand the contacts between the substrate and the enzyme in the ubiquitination reactions. So we asked the question about UBE2K. UBE2K is an enzyme that specifically forms K48 type chains. And so its K48 must be interacting with the active site. And so we asked how this UBE2K makes specific K48 type chains in the ubiquitination reaction. And we modeled this complex by Haddock, where we got the enzyme with the donor ubiquitin and the acceptor ubiquitin attacking the active site. And this is an in silico model that was done in Haddock. Depending on the model, we could see specific contacts between the lysine, incoming lysine, as I said, and on the active site, which is the 1692. We see a negatively charged residue from the E2, which is forming a hydrogen bond with the incoming lysine, and which is probably stabilizing the lysine to attack this active site. And when we mutated this D124, we see a rapid drop in rates of ubiquitination. The ubiquitination is almost non-existent. Compared to that, we had a lysine in here, which is also stabilizing the negative charge D124, which in turn is stabilizing the lysine 48. And when we mutate that lysine, we have also an effect on the ubiquitination rates, but the effect is much less compared to the direct substitution of this aspartic acid. There are other contacts around the lysine, which also are favorable contacts that probably stable this lysine to attack the active site between the Q49 and S85. And when we substitute the S85, also we see a rapid drop in the rates of ubiquitination. So this is the best model that we had, which we could validate using these ubiquitination reactions and monitoring by real time. There are other models which were not validated by this reaction. So you could have a variety of models and select the best ones depending on how the NMR experiments validate those models. We also asked how a different E2 pair like UBC13 and MMS2, which is a heterodimer, make this K63 specific linked ubiquitin chains, where the K63 is attacking the active site and forming this K63 specific diubiquitin. And this also we modeled in Haddock using all the different molecules present, the donor ubiquitin and the lysine, where the lysine 63 of the acceptor ubiquitin is attacking the active site. The contacts that we observed here also are slightly interesting. There is a aspartic acid D119, which is interacting with the K63 and stabilizing the incoming lysine K63. When we mutate the D119, we see a rapid drop in the rates of ubiquitination in real time. Then we also monitored the contacts between the other unit, which is the MMS2, which stabilizes this acceptor ubiquitin. So these contacts between the MMS2 and the acceptor ubiquitin positions the acceptor ubiquitin to attack for the lysine 63 to attack the active site. And when we mutate that, we had differential effects depending on the amino acid that was substituted. For example, if we substitute isoleucine 44, we see a rapid drop in the rates of ubiquitination. The other, also when we mutate a hydrogen bond in here between the S27 and the G47 backbone, we again see a rapid drop in rates of ubiquitination. However, when we substituted L46 V70 interaction, which is a hydrophobic interaction, we did not see, we did see an effect, but not a drastic one. So one could measure all of these interactions and their contributions to the rate of the ubiquitination by these methods. Now, I would also want to speak about the DUB enzyme. So since we can monitor assembly of ubiquitin chains, one could reasonably say that we could also monitor the disassembly of ubiquitin chains. And these DUB enzymes are the enzymes that disassemble the ubiquitin chains from the substrate. And this real-time NMR can also be used to do that, monitor that. To do that, we made this K63 linked diubiquitin, and we used a DUB enzyme which is specific to K63 linked diubiquitin. So this enzyme is going to cleave this bond in here and separate out the ubiquitin, acceptor ubiquitin, and the donor ubiquitin. So indeed, when we start the reaction at 0 minutes, we have a peak corresponding to this amide in here. And as the enzyme starts to cleave, the intensity of this drops, which allows us to measure the activity of the enzyme in real time by NMR. I would like to mention that we also did this using E. coli lysates where the MSH was overexpressed, indicating that it is also possible to probably use cellular lysates to do these kind of reactions. And the possibilities are endless now. One could monitor the ubiquitination reactions. One could monitor the inhibitors by screening against inhibitors if they inhibit the DUB enzymes, which will also show up in this kind of reactions. So these kind of NMR experiments can be used to observe a variety of interactions that are present in ubiquitination reactions. So in conclusion, we have three simple techniques to monitor ubiquitination real-time by NMR. The temporal resolution, I would like to mention that temporal resolution can be further improved by using non-linear techniques like BEST or something else. The technique can be applied to any other amine to amide post-translational modification. There are other post-translational modifications on lysines which change from an amine to amide, and those can be monitored as well. It is important to note that these techniques cannot distinguish between the different linkages between K6 and K11 and K63 and K48. These techniques do not distinguish between them. And we are trying to currently develop methods that will allow us to do that. While we are here, I'll just take a couple of slides to briefly mention one of the other results. So as we are trying to understand the host virus interactions, it is frequently observed that viruses mimic the components of the ubiquitin pathway. They have a minimal machinery, but they would critically mimic the ones that are very critical for their replication. For example, viruses are known to mimic the E2 enzymes. There are a lot of viruses which mimic E3 enzymes, and a lot of viruses which have their own DUBs or deubiquitination enzymes, including the SARS-CoV-2. So, but one of the interesting viruses are a nuclear polyhedrovirus, which is in the family of baculoviruses, and they actually encode for ubiquitin, which is the central component of the whole ubiquitin pathway. Now, why they encode for the ubiquitin and what are the differences of this ubiquitin is what we were interested in. But one of the major questions that we are also interested in, given that the host or the human cell has so many DUBs in it, there are about 100 DUBs in the human cell. How is the viral ubiquitin signal protected from this host ubiquitination enzymes? How does the virus ensure that its ubiquitin signal is protected from the host DUBs? Why does the host DUBs not cleave off these viral enzymes and inactivate the viral ubiquitin signal? So we saw the structure of the viral ubiquitin and compared to the host ubiquitin, there are slight differences between the structure, which could be interesting, which also changes the stability. But one of the interesting things is this viral ubiquitin has an additional lysine apart from the 7 lysines that I mentioned in the host ubiquitin. So this additional lysine we showed can be used to form this lysine K54 specific type of chains, and these chains exist in vitro, can be found in vitro as well as in cells. And what is most interesting is when we screen this K54 link chains against the dubs that are present in host, we see that these K54 link chains are resistant to the host deubiquitination enzymes. So the viral ubiquitin signal can be protected by creating a novel K54 type of link chain, which the host deubiquitin enzymes have never seen it and cannot cleave it. So we believe that this is one of the intelligent ways in which the virus ensures that its signals is protected, which will help it to replicate. I would like to start acknowledgement of the people who are behind this work. These are my students. They are the stars of my research program. The people who have contributed to the work that I spoke today have these bright rings on top of their head. Batul in here and Bazvi contributed to the work of the monitoring real-time NMR ubiquitination and simulation by real-time NMR. And Hitendra here is the person who's behind the work of the viral ubiquitin and the K54 type chains that are observed in the viral ubiquitin. I would also like to acknowledge the facilities and the financial support. We have wonderful facilities managed by wonderful facility managers, Purushottam, Nishant, Chaya, and I would specifically like to acknowledge Anil, who introduced me to NMR, Andy, who introduced me to biomolecular NMR, and Alan, who introduced me to the field of ubiquitin pathway. I thank you for your presence, and I can take questions now.
Okay, thank you, Rana. We have a couple of questions. Am I audible?
Yes.
Okay, there are a couple of questions which are in the chat box. First one is from Jitendra Chok. He is asking, in case of multiple lysines and multiple ubiquitination, how do you assign these new amide signals?
Yeah, so Jeet, that is a drawback of the current method. We were only looking at specific lysine side chains and E2s that make these specific lysine side chains. We are currently looking at ways to distinguish between the others. We have to use something intelligent to look at changes in the lysines, different lysines.
And, are you controlling mono ubiquitination levels or?
So these reactions are currently controlled in the diubiquitination reaction, and if you consider a substrate, yeah, that is the monoubiquitination reaction. Okay. But yeah, so I mean, beyond a certain length, you would not be able to monitor because the size would increase dramatically. Yes. But yeah, as long as we are interested in the specificity, like how are these specific chains being formed, or how is a branch being formed, then we can stop the reaction at a specific size, which is amenable for NMR studies. But still, we'll be able to ask these questions because the specificity, once we understand, is the same as long as the chain keeps elongating. So yes, it's the same. Okay,
for Jeeta, the second question about the size of the molecule, I think he got the answer.
Yeah. Second.
Okay. So another question by a person called Birdra. He's asking, do you use simple mixing or an in situ rapid mixing?
Oh, this one is simple mixing. Sorry, we did not use the rapid mixing, but yeah, that's something very that we are very interested in. We don't have the hardware to do it, but yeah, this we rapidly mix outside the NMR tube and put it in the NMR. So we lose, at about 5 minutes or so in this process. But yeah, rapid mixing would be fantastic. Yeah, we're looking for the hardware to do that.
There's a question by Dr. Grace. She is asking, is this the same rate at which ubiquitin happens in cell also, as it looks like a very slow process?
Yeah, so in the current reaction, in the conditions that we used, in the concentrations that we used tens of micromolars of E2 and around 1 micromolar of E1 or lesser than that. Yeah, it looks slower in that, but in this cell it is more rapid because I think most of the E2s in the cell are already conjugated, and so the ubiquitination happens slightly faster. But it would be nice and fantastic if we can now do this in cells and monitor it and see what happens.
Another question by person, he has registered by a number 821289. His question is the DUB enzyme specific to bond made by lysine side chain? Does it cleave other peptide bond?
No, the deubiquitination enzymes have a variety of classes. So some deubiquitination enzymes are specific to certain linkages. Some deubiquitination enzymes are specific to just any type of ubiquitin linkages, but they are specific to ubiquitin linkages between isopeptide bonds between the lysine side chain and another ubiquitin. Now, if they are normal peptide, they could leave normal peptide bonds, then I would say they would fall in the class of proteases and not ubiquitin proteases.
Okay.
However, so yeah, they specifically recognize the GG motif at the end of the C-terminal end of the ubiquitin, and this is why they are classified as ubiquitin proteases.
We have a couple of more questions, maybe one or two we can quickly take. For the others, you can rather send the answer by email or so.
Sure.
One is from Nikita Gupta. I may have missed this. How do we know that what kind of polyubiquitin chain E3 is going to catalyze and which kinetics we are measuring? It seems that all the lysines on the donor and the acceptor ubiquitins are available. My question being, how do we differentiate between K48 kinetics from K63 or say hybrid chains?
So in these cases, we have looked at specific E2s which make this, can I just go to the slide, which make this specific type chains. And so, yeah, our current study is restricted to looking the specific E2s. And as I am showing in here, the specific contacts made by this specific lysine with this E2.
Okay,
So
quickly, Himanshu Singh has asked a question. Could you comment on the relative ubiquitination rates for 7 lysine side chains? Which lysine is more prone?
Oh yeah, I have no idea, but so it's the ubiquitinations of 7 lysines we have not measured, and I don't know if anybody has measured it using a system, but I can tell you in this cell, when we observe, when people detected this ubiquitin type of chains by mass spec techniques, one would find the K48 and the K11 chains are much more rapid and much more prevalent, and the K63 chains as well. So K63, K11, and K48 are observed much more in the cell compared to the others.
So there's one question from Olumide. The question is it known how surface charge of ubiquitin are the portions which bind within the active site of E2, E3, and DUBs, etc., change based on the presence of homotypic, heterotypic, or branched chain presence?
That's a very good question. No, I don't think so. We understand it completely because apart from the lysines that we know, there are also arginines present in the ubiquitin and histidines. So these contribute to the surface charge, and there are long, I mean, one would expect that there are long range electrostatic interactions between this active site and these molecules, which sort of form these transient complexes probably, and that helps to specify the sort of specificity of these chains. There has not been any clear-cut answers to this question. I'm sorry. I wish.
Okay, I have 2 more questions. I think if you can give quick answer, I'll ask it. Otherwise, we may have to go to the next one. Very quick questions. You can give quick answers. Elvis Mortis is asking, in case of UBE2K, it is proposed that UBA domain, not found in other U2s, seem to have a role in poly-UB formations. Any way to confirm this using NMR methods?
That's a very good question. We are currently working on it. We are exactly working on this question of what is the role of the UBA domain on the UBE2K. And there's a lot of work done by Gary Shaw's group who have looked at it intensely. And according to their understanding, it does not play a role in the specificity of the ubiquitin chains because it is not a contact between, I mean, as I mentioned, probably the contacts at the active site is what is responsible. However, the UBA domain could be responsible for rapidly elongating these chains. And we have seen this for another E3, which also has a UBA type domain, which is Q domain. This is was done in Andy's lab. And there it helps to rapidly elongate these chains.
Okay, one more question, of course, I appeared again, but we'll ask the last question from Mona Lisa Swine. Can you please comment how do you selectively isotope labeled only one lysine?
Oh yeah, so no, so yeah, so that is probably a mistake in my explaining. So all the lysines are labeled here with the Anita N15. However, since, sorry, this is not the correct slide. This is the correct slide. However, since the E2s that we use make specific lysine chain, so only one of the lysines, although all the 7 lysines are labeled, only one of the lysines formed the bond and became an amide bond. And so we could observe that specific lysine amide and the others did not interfere.
Okay, thank you, Rana. There's one more question from Mahavir. I think we will email you. You can respond to him later because
Yeah,
for one
Mahavir
time
and
we
I
have to
talk all
stop.
the way, so we talk frequently. So
Okay, thank you very much. Okay.
Thank you so much.
Thank you.
all.
Professor Ranveer, you can stop sharing your screen so that, yeah.
Yeah, Ranveer, please stop sharing. Albert, you can stop.
So how do I share it now? One participant.
Okay, Vivek will help you.
Yeah.
You go to the green arrow things are down.
Yeah.
And then the share screen is there, you know.
Yes.
Thomas?
And then it says one participant can share at a time.
No, there are participants. You can try. It will happen.
I don't get my computer on this. I don't know why. Okay, there you go.
Got it. Okay. Share your screen and open your PPT. Okay, that's it. Okay, fantastic.
That's it.
I think you can share it now. Okay. So now let me introduce the second speaker of this today's webinar. This is Professor Albert Thomas. Albert is currently a professor of radiology, psychiatry, and biomedical engineering. And he is also the director of magnetic resonance spectroscopy at the University of California, Los Angeles. His research interests physics of MR imaging and spectroscopy, developments of new MR spectroscopic imaging techniques, evaluation of NMRS techniques in healthy tissues and in different pathologies. He has several awards. I'll read one or two. He was awarded in 2010, cover article of magnetic resonance in imaging, in medicine, October issue, echoplanar correlated spectroscopic imaging, implementation and pilot evaluation in human cough in vivo. And he has several authors in that. In 1991, he has been given Young Investigator Award, Vitacares Foundation Biomedical Engineering Grants. So now it is over to you, Thomas. You have next half an hour, please.
Okay. Good evening, good afternoon, and good morning to the American side. I would like to thank Surya Prakash and the organizing committee of NMRS for inviting me to this webinar. So let's move on. I would like to dedicate this talk to our past NMR colleague, Dr. Atreya, and my deep condolences to his family. So my today's outline, first I will talk about MRI basics and K-space encoding, single voxel spectroscopy, multi-voxel spectroscopy or spectroscopic imaging, And then we will go through different acceleration techniques, phase encoding, parallel imaging, echoplanar imaging, concentric rings, radial imaging, and more, and conclude. So this is NMR spectrometer. So the sample is kept inside a solenoid, and the yellow column is the sample that we are trying to get a spectrum. The RF is perpendicular to the main magnetic field B0 that everybody knows. So earlier 1D and 2D NMR had included main magnetic field B sub naught and radio frequency field B1. And then down the road in 1973, Paul Lauterbur, Kumar Welty Ernst, Peter Mansfield, they all used gradient based imaging on using an NMR spectrometer. Ralph Word from GE Medical had introduced gradient enhanced COSI in 1990. And spatio-temporal encoding, as many of you know, it has been used by Lucio Friedman from 2003 and onwards. Now, coming to MRI, it uses 3 magnetic fields. So the main magnetic field, like always, without that, there is no NMR. You have the gradient coils, the B0 gradient coils, and the RF B1 field created by this body coil insert. And the bore, free bore, we call it. It varies between 50 to now I think 70 centimeter they are making to accommodate obese people. So, so there are 3 magnetic fields, main magnetic field, and so we deal with like 1 Tesla to it's up to 11 Tesla right so far. This is on the MRI side. On NMR, I know you are more than double. The gradient fields, 1 to 4 Gauss per centimeter or 10 to 40 millitesla per meter. But right now we have gone up to 8 to 10 Gauss per centimeter or 80 to 100 millitesla per meter. So the radio frequency field excites or perturbs signals into a measurable form on the order of 0.1 Gauss, but in resonance with the MR signal. So, so there are 3 gradient fields, X gradient as shown here, and then Y gradient and Z gradient. So every imaging system will have 3 coils that can modify the static field length, field strength, and thus you have the control over changing the Larmor frequencies of nuclear spins along 3 directions. Okay, here comes the picture of how the coils are packed inside an MRI magnet. This is from Nishimura from Stanford. There's a book on MRI principles. So, you know, all the V0 coils, shim coils, the main magnetic field, everything is shown here. Okay, the first thing, so the effects of main magnetic field and applied slice gradient. So if you introduce the slice selective gradient by incrementing the field by 1 Gauss on both sides, so you see the resonance frequency changes from 63.83 megahertz to 63.87 megahertz. So that's the basic thing in order to map the spatial spread of the signal. So this is how we do that. Now I'm going to go through what Kumar Welty Ernst had shown in 1975. And what they did was they, like you do in 2D NMR, instead of Kumar Welty Ernst method had incremented this phase encoding duration, and that was modified by Edelstein et al. Keep the duration constant so that there is no differential T2 star weighting on the spectrum. And so you first fill the topmost grid, and the second grid, third. So you just increment the amplitude of the gradient from maximum to the minus side. Okay. So this is the raw K-space. So in NMR, you deal with temporal domain. Here it's a K-space domain. So there is a phase encoding, and then the readout during ADC is the frequency encoding. Okay. So KWE method is a method proposed as Fourier zygmatography, which was modified by Edelstein to spin warp imaging. So this is a spin echo MRI, and you see spin echo is encoded by phase encoding in the vertical. We call it posterior to anterior. And the readout frequency encoding is done between left to right, okay? And so the gradient applied along the y-axis will cause the spins to precess at a frequency determined by their y position, and it's called phase encoding. And next, a gradient is applied along the x-axis, and the spin echo is collected. The frequency components of the echo gives the information of the x position, and it's called frequency encoding. Okay, so that's the equation dealing with the data. And if you would define ky and kx and ky as gamma times the gradient amplitude times the duration of the gradient. Once you define that, the equation becomes rho of xy, and then if you Fourier transform that, you get the spatial information from each point in space. So this is a 2D MRI. So K-space, we don't call it FID or echo in the time domain. We call it in the K-space domain. So that's the equation I already showed you, and you can define this Kx, Ky in the two different axes. Frequency and phase encoding gradients control the imaging trajectory in K-space. So I just would like to point out a few nice books over the past 2 to 3 decades. And as you know, the technique was developed in the 70s, but the commercial scanners were released in the early 80s and so on. So that's the K-space data, the raw data. And day one in the early 80s, this was from a calf muscle where you see a coarse image. And now the technology has improved over the past 3 decades. So you get nice spatial resolution, distinction of different tissues. Okay. So MRI exploits NMR to produce water-based images. Signal from proton in water, grayscale caused by T1, T2 relaxation, and proton density within a voxel. So since this is used as one of two dependent parameters, that's why the increment of phase encoding was kept constant. Okay. So now we can go to 512 by 512. Voxel resolution in plane, sub-millimeter, great. Structural differences cause T1, T2 relaxation variation among voxels. Now, this has been a revolution in the past 3 decades. We are lucky to live in an era when MRI scanner was developed. Think about our parents and grandparents, and they had to be scanned using X-ray. So, but with all the anatomical imaging we have with MRI, It has multiplanar capability, nice soft tissue contrast. Anatomical MRI is largely limited to depicting morphological abnormality. Anatomical MRI suffers from non-specificity. For example, in breast cancer, brain tumor, any cancer, sensitivity is pretty good with MRI, but differentiating benign from malignant cancer is a major problem. So the question is, can something else add that information? So the underlying metabolic functional integrity of brain cannot be adequately evaluated based on anatomy. This applies to any part of the body. So to that end, there are several physiology-based MRI methods which have been developed to improve tumor characterization, diffusion-weighted or diffusion tensor imaging, in order to early diagnosis of cerebral ischemia when people get stroke. DWMRA is great, highly sensitive, and perfusion imaging is used after injecting contrast enhanced agent and get hemodynamic information. So MR spectroscopy is a great biochemical tool which can improve the overall specificity of cancer detection, and there are more applications. So this is Embden-Meyerhoff pathway. And you see all these chemicals appearing and then the end products. And so, you know, in addition to water, which is detected by MRI, spectroscopy is dealing with all the biochemicals, metabolites, and some of the end products of end terminals of proteins. And so the concentration is 1 to 10 millimolar or below. And so the sensitivity is a big issue. Now, let me show you how to get a single region of interest from a human tissue. This is like doing a non-invasive biopsy. So you localize 3 orthogonal slices, and then the intersection of that will give you a cube of interest or region of interest. So I'm just showing you here 5 4-millimeter thick slices resulting in 20 millimeter, and you can localize anywhere you want with a 2 by 2 by 2 centimeter cube, and which is shown here. So these are all received from the MRI on the MRI scanner. So we can localize this pink cube region of interest or volume of interest. Okay. What is the sequence that we use? This is nothing but a double spin echo. So you get first Hahn echo here, second Hahn echo. The only difference between MRI and here, you have to apply these 3 pulses along 3 perpendicular planes, X, Y, Z. So a slice selective pulse tips the magnetization into transverse, and then you get an echo of an echo. And this was shown by Bottomley. It was called PRESS, point result spectroscopic sequence. And there is also another version called STEAM, which uses stimulated echo, shown by Fromm in 1989. So you can place the region of interest in the occipital area, gray of the brain. You can also move the voxel. So once you have the MRI, the computer can calculate the coordinates, and you can get this single region of interest without any problem. And the drawback, each one takes 5 to 10 minutes. Nowadays, it's 5 minutes for each voxel. So if the doctor wants 4 to 5 different voxels, the time becomes 25 or greater than 25 minutes. So one application, this is in breast cancer. You see the dynamic curve after you inject the contrast agent, like dynamic contrast agent like gadolinium. And the problem is one type of this enhancement does not differentiate benign from malignant. So this is a 56-year-old malignant patient. when we did spectroscopy, we saw choline peaks, taurine, myoinositol, and all that. So that could improve the overall specificity. You don't see it in benign. So we showed this in 2010. And the second application, this is in brain tumor. So one of the problems with brain tumor, many patients come with a low-grade brain tumor, get treated, go home, and then they come back with a big tumor like a glioblastoma, and they die within 13 months. So there is a biomarker called 2-hydroxyglutarate, and we looked at the LCMS, mass spectrometry, after the biopsy was done, and the MR spectroscopy in vivo. So this is between mutation, brain tumor mutation versus wild type. The spectral characteristics are different. And we did it in a pilot group, and it was good. Now, the drawback of single region, it requires a large sample volume. The doctors would like to have it less than 1 centimeter cube or 1 ml. Requires many averages to get adequate SNR, limited coverage, so only one small region in one experiment. So now I move to how to get multiple regions of interest, which will be important for the doctor to know. There are multiple tumorous tissues or multiple suspicious masses. So the total experimental time, you want to make it around 10 to 20 minutes. So this is, just to give you an idea of in prostate, 40-year-old healthy volunteer. The prostate is shown here, and you see the spectral characteristics are different. The 3 distinctive metabolic patterns in periurethral tissue, central gland, peripheral zone, that's where 70% of prostate cancers are identified. So there is a greater coverage by spatially encoding time signals with phase encoding gradients. Phase encoded data is encoded in k-space. So more regions of interest can be sampled. How do we do that? So this is again double spin echo with one spatial phase encoding, like what you saw in Kumar-Veltians and spin warp method. So example, 3 vials, creatine, NAA, choline, and if you would apply the phase encoding gradients as shown here, these are the real imaginary domain of the signal, and these are the phase characteristics and Fourier transform along kx and the t. You get the spectral frequency versus spatial location. So this is 1D. Now, how do we do 2D? So we have to fill out 2-dimensional KT space. You can put the phase encoding gradients along X and Y, and each FID is phase encoded along 1 dimension, and shown by Truman Brown in 1982, Andrew Maudsley in 1984. So this is brain data where you can see multiple voxels of interest. So that's the frequency domain extracted from these, and the time domain. So the manufacturers have everything for you to look at it. This is the metabolite map of different chemicals. You can overlay this on top of the anatomical MRI, and it works great. So this is in brain tumor, and you know that Dr. Jagannathan and co, they have, and the Ames research group, they have done so much along brain tumor, prostate cancer, and breast cancer. And so this slide shows you a comparison between 3 Tesla and 7 Tesla magnet, whole body. And you see the brain image quality of different chemicals very nicely. Okay, how long does it take with the phase encoding? So if you do 2 spatial, 1 spectral, the total time is given by this equation here. 1 second TR, 32 by 32 takes 17 minutes. Now, if you do 3D MRSI, it will take 4.5 hours. If you do 32 by 32 by 16, 1 second repetition time. You can reduce the resolution to 16 by 16 by 8, 34 minutes, but the anatomical definition might have been lost here. Okay, so how can we reduce the number of excitations and bring the total time to less than 10 minutes? Selective averaging, parallel imaging, turbo spin echo or fast spin echo, echoplanar imaging trajectories, concentric ring trajectories, and I will come one by one soon. Radial encoding and total variation regularizer. Okay, elliptical weighting, reduce time. You get approximately a factor of 2. Parallel imaging, reduced acquisition also. You can also do average weighting, 1 average to the outer space. The central, you can do more averages, 8, 4, 2, and so on. So that reduces the time. This is one example of prostate cancer MR spectroscopic imaging. You see the T2 weighted MRI showing the cancer location. And so the peripheral zone is right here. You can see the multiple regional spectra overlaid on top of MRI. This is choline to citrate ratio image, and it lights up very nicely as expected from the T2 weighted MRI. Okay, there is also a multi-coil reconstruction, SENS and GRAPPA. It reduces scan time, shown by Prusman at ETH, Sarikson at the NYU, New York University. Advantages: reduces scan time. Drawbacks: reduced SNR from reduced number of excitations. Okay, so first, echoplanar spectroscopic imaging. This was shown by Peter Mansfield in '84, and Stefan Passe from Switzerland, who was a student of Walter Aue. In '94, he showed the first whole body echoplanar imaging, and in my group in 2008. Okay, so we use repeated bipolar trains to read out positive, negative, 256 times or 512 times. And 2 sets of echoes, odd and even, and which are mirror images of each other. So you have to deal with 2 spatial, 1 spectral. So if you Fourier transform the temporal domain, you get a nice spectrum, and the spatial information can be Fourier transformed also. So in echoplanar spectroscopic imaging, a single line in the K-space is acquired in 1 shot. And the total time is nx times tr times nex. You remember with phase encoding, it was nx, ny. So an order of magnitude is saved by echoplanar imaging. And that's how they do functional MRI in less than a minute or less than a couple of seconds. And so the total time, again, so I already went through for the phase encoded. So with the echoplanar spectroscopic imaging, with the 32 by 32 16 resolution, it takes 8.5 minutes. So with the 3D phase encoded, I showed you already, it took 4.5 hours. So there is a significant reduction here. Now, drawbacks: echoplanar readout creates undesired eddy currents, which can distort spectral quality. Reduced SNR from reduced number of excitations, and switching the B0 gradients on and off multiple times, like 512 times, can put a heavy stress on the system. So the next one is concentric circular sampling. So in this rectangular Cartesian grid, so you need 16 by 16 shots, whereas with concentric rings, you can do it with the 8 rings. So there is a saving of time by a factor of 2. The outer corners of K-space contain very little signal. The main signal is coming from the central region. You know, this was shown in 1986 using rotary echoes, using RF field. Paul Lauterbur and co. there, sorry, it's 1998. And Holden Wu at Stanford in 2009. They had used concentric ring trajectories. And in my group, John Priyama in 2012. So a single ring in K-space is collected in a single shot. Imaging data cannot be processed by 2D FFT since it is not Cartesian, it is in the polar space. The total acquisition time is, so it's a 50% of what we saw with the FC. And so how do we design the gradient waveform? And it is shown here. So the equation for Kx and Ky, those are the two equations. And how do you program the gradient pulses is shown here. So that's the pulse sequence. So you repeat these sinusoidal, cosinusoidal gradients to get the temporal domain and the Kx, Ky, if it is a 3-spatial Kz also. So this is how the concentric ring trajectory goes. The central ring, innermost ring has the higher sensitivity. And you cannot do FFT, so you have to use convolution of different points into a Cartesian grid, shown in these references, 91 and 98. Okay, so in a phantom, just to show how the polar space data is lined up with the 8 rings, and we did it in human brain. You see the concentric ring trajectory spectrum opposed to the echoplanar spectroscopic imaging, just for comparison. So the total time again, so this is the fastest so far in this race. So the brain coverage, you know, recently, this is in 2020, investigative radiology from Austria, they showed with the 32 by 32 by 31 matrix, you can go to 3 minutes, and with this kind of resolution, 15 minutes. So with Uzay Amir and co-workers, recently they showed with the 3 minutes with a concentric ring, you can get it. Sorry. So advantages are shown here, and the drawbacks. SNR gains from averaging compensate so that sensitivity per time in both sequences is similar. And data must be regraded, which you know, there are different papers which show how to do it. And the last topic, how can we accelerate more? You can use projection reconstruction using radial encoding. Series of projections taken at different angles shown by Paul Lauterbur. But what we did was, so this is the Cartesian grid, and the B is radial encoding, and C is, there is something called golden angle radial projections by 111-degree separation. And we can also do undersampling and TV reconstruction. So quick brain, just 1 second. So with the radial, you know, this is the brain data, and you know, the brain image from this radial encoding, and extracted spectra at 4 different locations. The locations are shown here. So this paper is right under being revised right now. So you can bring this down to three minutes or so. And so I just wanted to point out to you, MRI started from all the developments happened in the 50s and 60s from NMR. And in the 70s, Lauterbur, Kumar Welke-Ernst, Raymond Damadian, and Peter Mansfield, they did so much of work in the 70s, which led to the revolution in our lifetime. And so spectroscopy in vivo, this started in the 80s, and it's still improving. And now I see NMR field also has borrowed all these projection reconstruction, ultra-fast 2D NMR with the spatio-temporal encoding, and how to use compressed sensing beyond the Fourier transform by Mobley and Hogue. So we get benefited on both sides. So we started from NMR, but there are some techniques which are going to NMR too. Okay. So this is an MRI scanner with different RF and the magnet and all the other things you need. Okay, in conclusion, as a physicist, as an NMR person, it's a great experience to work on MRI, which has been a revolution in medicine during our time, thanks to NMR. MRI sequences can be easily translated into MR spectroscopic imaging. All these sequences have been implemented 3 Tesla, 7 Tesla, 9.4 Tesla, and there are more than 10 Tesla right now being tested. Accelerated polar and radial MRC data and regrading to Cartesian. And the question is, once we get down to less than 5 minutes, functional MR spectroscopy, how the metabolites change in real time can be recorded. And so the multi-dimensional spectroscopic imaging from my group using acceleration and nonlinear reconstruction using compressed sensing. And, there could be 60, 70 down the road, which is certainly possible. And I like to thank all my coworkers, current and all the past students, past postdocs, and my outside collaborators, Sar Ramadan, Uzay Amir at Purdue, Yasmin Hurd at Mount Sinai, and the 2 funding institutes for supporting this work. And yeah, one take-home message, MRI and in vivo spectroscopy, we apply this in human diseases. And initially, I did not like to be in a hospital environment, but as a physicist, I am accepted into medicine. There is a medical physics field, and there are a lot of opportunities. And so I like to thank you all for listening to my talk. Thank you.
Thank you very much, Albert. We have a couple of questions, which is in the chat box. One is from, I think it is Vivek. What is your take on frequency drift brought by B0 drift during the long acquisition of 3D, 2D echoplanar imaging? And how do you account for B1 inhomogeneity issues for 3D EPI at high fields?
Okay, so B0 drift, there are several papers addressing this because in a 50 centimeter bore magnet or 70 centimeter bore magnet, B0 can drift significantly, and there are ways to predict this B0 drift in different locations. So you can do a reference scan using water. Water is highly abundant, 100 molar. And using this water scan to correct for B0 drift in, with the metabolites. So that can be done. And the second thing is how the B1 inhomogeneity is addressed. My answer to that is, there are adiabatic pulses which deliver homogeneous B1 field. So they become longer RF pulses than normal, sync pulses or whatever. So, but this has been addressed using adiabatic pulses from year 2000 and onwards. So both issues are answerable. Are we perfect with that? I'm not sure. We are still improving. Yes.
Okay, Thomas, there is another question from Dr. Durgesh Dwivedi. He says it's always great to hear MR physics from Albert. Consider the challenges of MR spectroscopy, such as long acquisition time, post-processing, and a few controversial results. What is the future prospect of MRS or MRI in breast and prostate cancer? Thanks.
Yeah, I would like to clarify there is no controversies. The standard deviation you get in healthy control. So you take each metabolite, mean and standard deviation. Can you make the standard deviation 5% of the mean, or you end up with 40% of the mean? Because there are diseases where there will be the metabolic changes within 5% or 10% of the mean value for each metabolite. So, but the scanner sensitivity is improving. Now we are dealing with 9.4 Tesla. You can use homogeneous B1 field using adiabatic pulses. V0 drifts can be kept under control. So, there are still a lot of challenges, and that's one thing I enjoy working in imaging. The field is improving day by day. And compared to my days in the late 80s, and when I look at now, there is so much of improvement on the hardware side, on the computer side. So the total duration, as I just showed you, you can. So what the doctor wants to see is, can you finish it in less than 15 minutes? If they do 45 minutes of MRI, so in 15 minutes, can you do spectroscopy? So that's why we are dealing with all these acceleration techniques. And so does that answer?
Okay, I think so. I hope. Yes.
Can I have a question, NSP?
Yeah, I'll come back to you. Subbu, I'll come back to you. I have a couple of questions here.
Okay.
Definitely, I'll come back to you.
Okay. All right.
There is one question from Rajat Gar. He's asking, what are the good books to study MRI from a methodology perspective? I think this answer can be sent by email. There is no need to answer here. And Himanshu Singh,
slides also has it, yeah.
I'll send you the question. You can give him the reply. He wants some books on MRI. Could you comment on the brain water lifetime? This is from Himanshu Singh in healthy versus disease states. Brain water lifetime in healthy versus disease states.
Brain water lifetime. Brain water is always there. In tumor, there is angiogenesis, there is more water elevation. And whereas in, if all of us are healthy, our brain water is 75% of the tissue of brain or any other tissue. So that will be my answer, yeah.
Okay. There is a question from Dr. Nitin Lobo. How do you compensate the physical movement of patients during the measurement?
Okay, so if we succeed with the radial, for example, you can minimize the motion-related artifacts. We have tried it in liver, kidney, and so you get some help. But you train children, young humans, we educate them how to be inside an MRI scanner without moving the head, without moving your head, body or legs, if you are dealing with the calf muscle, thigh muscle. So motion is a real problem. And, you know, so we try to keep it under control as much as possible. Yeah.
Okay, I think there's another question from, I think, Vivek again. What is the best spectroscopic voxels resolution that is achievable within good gradient strength limits?
So right now we have like 8 Gauss per centimeter or 80 millitesla per meter. So on NMR magnets, I know you have more than 10 Gauss per centimeter, maybe 50 Gauss per centimeter. Okay. So in our case, I would say, you know, 0.1 centimeter cube. Okay. So that's the 3 Tesla and 7 Tesla. 7 Tesla could be better, and higher field strengths down the road. So in the next 10, 20 years, this will change. You will get better resolution. Yeah. Okay, I think we'll come back to a question which Subramaniam wanted to ask. Professor Subramaniam, your question, please.
As far as this spectroscopic image is concerned, considering the large voxel volume and fairly poor SNR, people are now looking into hyperpolarization by increasing sensitivity by 2 or 3 orders of magnitude. This facility is not available everywhere. This is fairly specialized technique, but with UCSF, they have done hyperpolarized MRI metabolites in pathological prostate cancer. All the imaging is done within a matter of a couple of minutes, and with resolutions which are of the order of 2 millimeter cube. That. So, is there any effort to increase the sensitivity via either hyperpolarization, or is it too expensive a technique? Or what is happening?
UCLA, my chair hasn't agreed to get a hyperpolarizer. As you know, UCSF had started in mid-2000. It's 15 years now, and there is no real breakthrough. Okay. For radiologists, they want to see how can I apply this to humans. And so until we start seeing new agents instead of pyruvic acid with the carbon-13, can we come up with other contrast agents which will last longer and better sensitivity, better specificity? I think it's an ongoing research, Subbu. So we have to wait and see, not now. I'm not doing hyperpolarized, unfortunately.
Yeah, it will, Albert, it will take some time to come to clinics because of the short lifetime. So except for that one particular paper on the prostate with the pyruvate, there are no clinical evaluation till now.
Yeah. Yeah, that's right. So, certainly, the issues with, when you deal with the medical doctors, you have to have a sellable technique for them. Otherwise,
yeah, it will take little time mainly because of the lifetime and the practicality attached to it.
Compressed sensing, maybe a little bit more compressed sensing and reduced case base kind of attempts will reduce the measurement time for a given
yeah, that's right. 5 to 10 minutes with acceleration and compressed sensing. That's what my group has been working on. And with the radial, maybe faster than that. So, these are ongoing projects which I'm sorry, we are still not 100% there like MRI, but this is an ongoing process, and I'm happy that I'm able to come up with a new spectroscopic imaging techniques. And I think with all my training from IISc Bangalore with Professor Kumar and Professor Richard Ernst at ETH, I think there is a great opportunity. This is an evolving field, and as long as there is more opportunity, I can survive. Otherwise, I lose my job.
Albert, can I add one thing to the question that how do you compensate for physical movement? If you don't mind, there are nowadays all the new MRI scanners in the last 4 to 5 years come up with a post-processing algorithm which will take into account the movement of the head up to, I think, if I am correct, up to 30 degrees on both sides.
Yeah.
So that's a very, very big improvement for neonates, children who cannot be held inside the magnet for a long time. Yeah, I think
You
this
know,
is need
still,
to.
on the MRI side, things happen very quickly because water concentration is, 100 molar, 100,000 millimolar. Spectroscopy, we are still dealing with 1 to 10 millimolar. So the cryo probes are yet to come into whole body, and so more things need to be done, in order to improve.
Yeah, I think it's a very interesting topic. There may be a lot more questions people want to ask. I think for want of time, we will stop at this. Thank you, Albert, once again for your excellent presentation.
Lovely presentation. Yeah.
Okay.
You're welcome.
And
Thanks.
now I request Vivek to show the slide of the next Zoominars of next week, speakers for next week. Vivek, are you there? Vivek? Or Jeet, one of you.
Yeah, I'm doing that. Actually, it was there.
Okay, fine. So these are our speakers of next week. Of course, Vivek himself and Nagan Gowda is Nagan Gowda from Seattle. These are the speakers. Okay, thank you. We will see you again next week.
Thank you, NSP.
Thank you.
Thank you.
Thank you.
Thank you.
Thank you.
Thank you.
Nice series.
Thank you. Good night, everybody.
Night.
Good night, everyone.
Oh, that's a nice picture.
Anil, are you talking to me?
Yeah, no, it is a nice lecture by you.
Thank you.
Mm.
Yeah, lately, I think the last decade or so, there are a lot of improvements, progress. So it's good to be, working on pulse sequence development and so on. Yeah.
Thank you so much, Professor Thomas. It was really instigating talk. Thank you.
Oh, thanks a lot, Jeet. I forgot to thank Jeet.
Thank you, Professor Thomas.
Thank you. Bye.
Yeah. Bye, everyone.
Bye, Anil. I will.